So if you were here last time, you heard me speak about the case for intelligent design, looking at irreducible complexity and basic structure of the argument for design. And today I want to talk about the molecular challenge to evolution. Now I'm going to start with a quote from Richard Dawkins, who wrote that it's absolutely safe to say that if you meet somebody who claims not to believe in evolution, that person is ignorant, stupid, or insane, or wicked, but I'd rather not consider that. Now, hopefully I'm not ignorant. I have a PhD in the subject of evolutionary biology as well as a master's. So that leaves either I'm stupid or insane or wicked by Richard Dawkins' understanding. Now, hopefully, what I hope to show you over the course of this presentation this morning is that you can actually make some very sophisticated and solid scientific objections to evolution, which are not based on interpretations of religious texts or anything like that, but are purely based on empirical, scientific, observational evidence. I'm going to go through this morning 3 scientific challenges to evolution. There's many more that we could talk about, but I'm going to talk about 3 of those for the next 40 to 45 minutes. Those are the waiting time problem, the problem of reductive evolution, and the origins of animal body plans. Next week, I'm planning to talk about the origins of life and the origins of protein structures and that type of thing. So let's talk about, first of all, the waiting time problem. Now, if you were here last week, you heard me talk about the argument from irreducible complexity. We talked about how the cell is chock full of nanotechnology and irreducibly complex systems, that is, systems which are functionally coherent. There's a high-level objective being accomplished by numerous subcomponents which are performing subfunctions, which all contribute to the system performing its job. And if you removed one of those components or 2 of those components, the system would not work half as well as it used to, or a quarter as well as it used to, but it would be broken. And so what I want to discuss for the first part of this presentation this morning is, can we mathematically express exactly why irreducible complexity is a challenge to evolution? And this is what we call the waiting time problem in evolutionary biology. So I want to start by defining what we mean by coordinated mutations. So coordinated mutations is when you have 2 or more coincident genetic changes that only together produce an adaptive phenotypic effect that allows for selection to operate. So we talked last time about how DNA gets copied by an enzyme called DNA polymerase, and sometimes, although it's highly accurate, sometimes it makes a mistake, and we call those mutations. And when we're trying to account for systems which are irreducibly complex, then you need mutations to occur that need to occur in a coordinated way, because one of the mutations on its own isn't going to get you the system, but it's only when you get multiple changes happening simultaneously in a coordinated way that some adaptive benefit is realized, okay? So what then is the waiting time problem in evolutionary biology? Well, evolution is supposed to proceed by random mutation and natural and sexual selection. That's the non-random component of evolution, that natural selection and indeed sexual selection preserve the successful gene variants, those gene variants that help the organism to reproduce, and those successful combinations of gene variants end up, the advantageous ones end up becoming more dominant or more frequent in the population over time. Now, selection can only work on mutations with a positive or negative adaptive value. So there's different types of selection. There's positive selection, there's purifying selection, and those select or purge from the genome those mutations, those changes which are either advantageous to the organism's survival or disadvantageous. And at least some adaptive advantages, as I hope I showed last week, require 2 or more coordinating mutations in order to be realized. Now, all mutations have 2 time constraints that depend on the population size and the generation time. Those are the waiting time for a mutation to occur in the population, right? So this is how long is it going to take for chance mutations to produce a particular mutation in the population? And the waiting time for the fixation of this mutation. So it's not just enough to get a mutation. It's not just enough for that gene variant to arise in the population. It's got to go from being the lone individual out there to becoming the norm that gets established in the population. And that's known as fixation. Now, the question that we can ask is, does the history of life provide the resources for evolution to accommodate these waiting times? And I'm going to answer that question decisively in the negative. So the waiting time dilemma then is as follows. With large populations, the waiting time for a mutation to occur decreases, but fixation time increases. Now, why is that the case? Well, if you have a large population size, then there's more individuals for a mutation to arise in. So you have more probabilistic resources, if you will. So the waiting time for the mutation to arise in the first place decreases. But the time for that mutation, that gene variant to become fixed in the population increases because there are more individuals for it to sweep through, right, in the population. On the other hand, with small population sizes, the waiting time for a mutation to occur increases, but fixation time decreases because you have fewer individuals for the mutation to arise in, so you have less probabilistic resources, so it takes longer for the mutation to arise. But then on the flip side of that, the fixation time will decrease because there's fewer individuals for that mutation to sweep through. So thus, there's no easy way for evolution to work around the waiting time problem. If with a large population or a small population, you either have to contend with the waiting time for the mutation to arise or the waiting time for the mutation to become fixed in the population, to go from being that lone individual out there with that mutation becoming the norm that gets established. Now, for those of you who are technically minded, I'll just say briefly counter one popular response to this, and then I'll move on. So one popular response to this is that recombination allows for neutral mutations, and neutral mutations are about 75% of all mutations to occur separately in a population, and to combine later by sexual recombination. There's a paper, however, in 1998 published by Christensen et al., and they showed that recombination lowers the waiting time until a new genotypic combination first appears, but the effect is small compared to that of the mutation rate and population size. So it doesn't really help you that much. Now, let's do the math though. While the fossil record provides the data for the available time frame, the standard formula of population genetics allows us to do the math. Now, all you need are reasonable estimates of the following three parameters that can be established by comparison with recent organisms. You need the mutation rate, the effective population size per generation. Now, the effective population size is that population of individuals that reproduce, right? The others that don't reproduce, you don't factor in. And then the generation turnover time. And so this problem was first discovered by Michael Behe and Snoke, David Snoke from University of Pittsburgh, and Michael Behe from Lehigh University. So Behe and Snoke published a paper in 2004, and then Michael Behe published his book, on the Edge of Evolution, 2007. And that he made the argument that the waiting time for two coordinating mutations is prohibitive for the neo-Darwinian mechanism of evolution to work. Behe used the example of malaria resistance against the chloroquine, required two mutually dependent mutations, applying these data on human evolution, predicted the waiting time, and that should say of 10 to the 15 years. So it's 1 with 15 zeros after it. Now, in 2008, in the Journal of Genetics, there were two mathematicians called Rick Durrett and Dina Schmidt, and they attempted to respond to Behe's waiting time problem. And they said, no, it wouldn't take 10 to the 15 years for a mutation to arise. Michael Behe's data had been based on empirical observations from malaria, but they put forward a mathematical model in which they said it wouldn't take as long as 10 to the 15 years. It would only take 216 million years for a two-step coordinated mutation to arise in a hominid lineage. Now, bear in mind that until very recently, the breeding or effective population size of hominids has only been about 20,000 individuals on average per generation. And so they, based on that estimate, they put forward the mathematical model and said that to get two coordinated mutations, which are only adaptive by themselves, when they're only adaptive when combined, not by themselves, it would require 260 million years for that to arise in a hominid population, in contrast to Behe's earlier estimate based on empirical data that it would require 10 to the 15 years. Now, obviously that's still prohibitive because the human chimp divergence, according to most estimates, is somewhere between 6 and 8 million years ago. So given that you need 216 million years for a two-step coordinated change to occur, that seems still rather prohibitive. Now, there was a subsequent paper published in 2015. This was by John Sanford et al., John Sanford from Cornell University. And he used a computer simulation to calculate the following waiting times based on reasonable estimates for an ancestral hominin population of 10,000 individuals and a generation turnover time of 20 years. And he calculated for the fixation time that the fixation of a specific point mutation was between 1.5 and 15.9 million years, and the fixation of a single codependent mutation, 85 million years. So you've got the problem of the waiting time for that mutation to arise in the population in the first place, and then a second waiting time problem for that mutation to become fixed, to go from being that lone individual out there to becoming the norm that gets established in the population. And that's very prohibitive. But it gets worse, because changes in amino acid sequence to give a protein a new function must often be accompanied by corresponding changes in regulatory sequence. So for example, it's thought that hemoglobin and myoglobin are related, and hemoglobin, as you probably know, is the protein which is responsible for carrying oxygen around in the blood, and myoglobin is a protein which deposits oxygen to the muscles and tissues where it's required, and its a function is made possible by its stronger binding affinity for oxygen. And it's thought that at one point, the ancestor of those genes duplicated, and one copy retained its original function of carrying oxygen around the blood. Myoglobin was then free to go and carry out this other function of depositing oxygen in the blood. But myoglobin, if it was present in red blood cells, would not provide a selective advantage. In fact, it would be harmful to the organism because it would bind too tightly to oxygen and not release it to the tissues. And so the changes that are necessary to transform something like hemoglobin into myoglobin need to be supplemented with coordinated changes in the regulatory sequences to ensure that it's expressed in the right place. Now let's look at a case study in whale evolution, because what's really nice about the fossil record is that the fossil record often gives us a handle on the probabilistic resources that are at our disposal in terms of the population sizes, the generation turnover time, and the mutation rates. These can be established, and then the fossil record gives us a time window for an A to B transition to occur, right? So what sort of changes do you need to transform a land-dwelling terrestrial mammal that looks somewhat like a deer called Pakicetus to a full-blown sea-going mammalian whale, right? So the series of the whale series in the fossil record is thought to be a very is often held up as a flagship example of an intermediary series, a transitional series of forms, going from Pakicetus to Indohyus to Ambulocetus, which is a walking whale, to Protocetus, all the way down to Dorodon Basilosaurus. And the most generous estimate in the literature for how long that transition took to occur is approximately 9 million years, although you can make a case that it took far less time than that. Now, what sort of morphological changes, anatomical changes would you require to transform a land-dwelling terrestrial mammal like a deer, called Pakicetus, into this full-blown sea-going whale? Well, first of all, you need an intradominant current heat exchange system, because unlike in most mammals, in whales and dolphins and porpoises, the testes are actually inside the body, right next to the muscles that are generating heat during swimming. Now, in most mammals, what happens is if the testes fail to descend and they're not cooled, you get sterility, which is obviously an evolutionary dead end. And so you have to cool down the gland. And that's done in a manner akin to a modern refrigeration system by shunning cooled blood from the tail flukes down to a nexus of arteries and veins that dissipates the heat from the gland. And so even though the cetacea, the whales, are moving about in the water column, the testes remain below core body temperature. So that's an elaborate innovation that needs to be accomplished in order to facilitate that transition from the land-dwelling deer-like creature to the aquatic whale. Another thing is the reorganization of kidney tissue to facilitate the intake of salt water. You're no longer drinking just fresh water, you have to intake salt water. So that requires a reorganization of kidney tissue. You need a reorientation of the fetus for subaquatic birth because you're no longer coming out head first. Your fetus is coming out tail first. Modification of the mammary glands for nursing young underwater. The forelimbs have to be transformed into flippers. You have to have substantial reduction of the hind limbs and the pelvis. The tail has to be transformed into a fluke, including a ball of vertebra for up and down movement. Unlike a sacral vertebra, which moves from side to side like a dog's tail, now it has to move up and down. So you need a ball vertebra. And you need a special lung surfactant because when the whales dive and come back to the surface, the lung has to re-expand and do so very quickly. Now, let's just assume that let's just say one of those examples, take the intra-abdominal cardiac system that we talked about previously. Let's just assume, for the sake of argument, and I think this is an absurdly generous assumption, that only 2 mutations are necessary to accomplish that change. So let's say one mutation to get the testes inside the body and another mutation to get the elaborate cardiac system that I described. Well, how long would you require in order for that to take place? So we already talked about how in the Dritten-Schmidt paper, which is based on the hominid population, it would require approximately 216 million years according to their mathematical equations. Now, Richard Sternberg, he was an evolutionary biologist based in Washington State. He basically took the equations from the Dritten-Schmidt 2008 paper and he applied it to the population of terrestrial mammals to account for we're dealing with more individuals and so forth. And according to his math, for the whale series, it would take approximately 43.4 million years for that 2-step coordinated mutation to arise. But according to the most generous estimate from the fossil record, for the entire transition from Pakicetus to Dorodon and Basilosaurus, you only have about 9 million years. And in fact, according to some estimates, you have far less than that. So the math just doesn't add up. We can also look at bacteria because bacterial populations are much more massive than animal populations, and they have much faster generation turnover times and much faster mutation rates. And so you can actually model millions of years of human evolution by looking at bacteria because they have such rapid generation turnover times and such massive population sizes. And so if this waiting time problem is a problem in bacteria, it's all the more a problem in animals such as humans. So this is a paper that was published some years ago in the journal Biocomplexity by Douglas Axe. And he basically put forward a mathematical model of population genetics for a bacterial population. And he says, this is a quote from the paper, since we now have a large and rapidly growing catalog of functional protein systems that seem to be fundamentally complex, there is a growing sense that innovations of this kind would require complex adaptations, meaning adaptations needing not just one specific new mutation, but several, with all intermediates being non-adaptive. If so, this may present a probabilistic challenge to the standard evolutionary model because it would require fortuitous convergence of multiple rare events in order for a selective benefit to be realized. And this is a figure from the paper. And basically, the purple line is representing his estimate for, so this is assuming a gene duplication event where the gene duplication event itself is neutral, so it's neither advantageous nor disadvantageous. In this case, this blue line, it shows that you require that once the innovation, the adaptive novelty requires more than 6 coordinated mutations, it's improbable to arise by chance. However, if the initial duplication event was maladaptive, meaning it's disadvantageous to the organism's fitness, then the maximum it can require is 2 or fewer. If you look at this purple line here, it's 2 or fewer. So that's especially prohibitive when we're dealing with some of the systems we talked about last time, like the origins of bacterial flagella or bacterial DNA replication and so forth. So let's look at a second problem, and that is the problem of reductive evolution. Now, the Darwinian model of evolution holds that one of the key mechanisms of evolutionary innovation is the duplication of genes and the subsequent divergence of one of the duplicate copies to undertake a new functional role, like how I described earlier with hemoglobin and myoglobin arising from an ancestral gene. And now, because the probability of a single gene stumbling upon a significantly different yet functionally advantageous sequence is so small, the idea is that following a duplication of a gene, one copy is able to retain the original function, while the other is free to explore the vast sea of combinatorial possibilities in search of some novel function. And it's widely believed that a duplicate gene has no phenotypic cost, no cost to the organism or advantage that's associated with it. That is, it is selectively neutral. And in such a state, it's thought that the gene is free to mutate independent of selection constraints or pressure. So when a previously protein-coding gene incurs deleterious mutations such that it's no longer codes for a useful polypeptide, that the gene is referred to as a pseudogene. So it's now a broken gene, and it's able to explore combinatorial space, sequence space, in search of some new function because it's freed from selective constraint. Now, here's a paper that was published back in, I think, 2010. Yeah, 2010 by Anne Gager and Ralph Silke and others. And they demonstrated that the process of reductive evolution was sufficient to prevent lines of E. coli from taking a simple two-step pathway to a new fitness function. So in this study, there was a gene that's known as TRPA, which is one of the genes that codes for an enzyme that's involved in the biosynthetic pathway, leading to the synthesis of the amino acid tryptophan. So this gene was broken in such a way that it could recover the ability to synthesize the amino acid tryptophan by reverting only 2 single point mutations. So one of the mutations was partially inactivating of that gene. The other mutation was fully inactivating of that gene. So you would think that that's a very easy adaptive pathway for natural selection to take, right? That one mutation would gain back part of the function of that gene. The other mutation would gain the full function. So you have a nice selective pathway going from no function to partial function to full function, something you would think that it would be quite easy for the evolutionary mechanism to find. The authors here concluded that the cost of transcribing a broken gene incurs a significant fitness cost, and thus its removal is facilitated by positive selection. So there's actually a fitness cost associated with carrying a broken gene, especially in bacteria, where there is a high selective pressure for small genome sizes. And so carrying a broken gene actually carries an energetic cost because of the energy involved in transcribing or expressing that broken gene. So the gene and all of these lines of E. coli, basically, instead of taking that two-step adaptive pathway, natural selection actually just got rid of that broken gene. It was purged from the genome. It incurred more deleterious mutations to stop it from being expressed at all. Okay, so another paper came to the same conclusion later that same year in 2010. This is a paper in the journal PLOS Genetics called The Extinction Dynamics of Bacterial Pseudogenes by Keogh and Ochman. And they examined the genomes of Salmonella species, arguing that the evolution of bacterial pseudogenes, broken genes, is not an entirely neutral process. To the contrary, bacterial pseudogenes are often actively degraded by the forces of selection because of the energy cost of transcribing a broken gene. So the degradation of genes, which do not code for proteins or perform regulatory functions, is most probably due to the energetic cost of transcribing them into messenger RNA. So again, coming to the similar conclusion that there's a time constraint on the time available to search through sequence space, because the gene over time is going to incur mutations that totally inactivate it. So that is the problem of reductive evolution in bacteria. Okay, let's come to a final objection to evolution, and this is the origins of animal body plans. Now, here we see and this is the fertilized egg, or the zygote, the single cell, which and this is the evolution. This is the developmental trajectory leading to the adult form capable of reproduction. In this case, it's a roundworm, or Caenorhabditis elegans is its scientific name. And ontogenetic depth was a concept that was coined by Dr. Paul Nelson, who is a philosopher of biology. And it refers to the distance for any animal between a single-celled state and the adult capable of reproduction. And basically, in order to in order to get from this fertilized egg or this zygote to this adult form capable of reproduction, you have to traverse this distance, this ontogenetic depth. And basically development takes place by a process, rounds of cell differentiation and specification, where you have your zygote that differentiates into 2 daughter cells, and then those differentiate into 4 cells and so forth. And at each stage, with each round of duplication, you're down-regulating certain genes, not with respect to what those cells are going to be doing, but with respect to what their descendants are going to be doing. And so you have to have a goal in mind, an objective, a higher level objective that you're leading to. And natural selection isn't able to visualize complexity and bring things together that are necessary to realize a complex end goal. Natural selection is blind to the future. And notice if you traverse only halfway across this distance, or even 3/4 of the way across, you don't have a halfway live roundworm. You have a dead roundworm. And so how do you put together a developmental trajectory like this? By a process of natural selection, preserving some level of function all along the way. I'm going to show a short animation. I'm just going to I'm just going to show a short animation that shows the process of embryonic development in a roundworm, and highlight some of the problems facing the evolutionary narrative in regards to explaining this sort of phenomena.
You can find them in the soil in your backyard, but they're so tiny you will need a microscope to see them well.
C. elegans is a very small, about 1 millimeter long nematode. It's a roundworm that you can isolate from a compost heap, and they consume bacteria there, and they're all around us. They're members of a very large phylum, has hundreds of thousands of species, called Nematoda.
C. elegans is one of the simplest kinds of animals known to man. But in reality, it's not very simple at all.
We call it simple because we happen to be a lot bigger, but 100 million base pairs of DNA is not simple.
In 2002, 3 scientists were awarded the Nobel Prize for Medicine for their work unraveling the mystery of how C. elegans develops. One of those scientists, John Sulston, meticulously documented the building of the worm from its first cell division to the final adult form. Sulston's research opened an extraordinary window into the cellular processes required to construct even a relatively simple animal.
For the first time in the history of biology, we were able to see and track the development of an animal from one cell to the adult. It had never been done before. So really, for the first time, biology was able to say, here's how you build an animal via development. That kind of knowledge is very precious. To have that, and to have that in the detail, right down to the level of individual cells, that's incredible. That's definitely worthy of a Nobel Prize.
Like nearly all animals, the process of building C. elegans starts with a single fertilized egg cell. That single cell divides into 2. Those 2 cells divide into 4, which divide into 8, which divide into 16, and so on until there are around 1,000 cells. But the final cells in an adult C. elegans are not the same as the first cell from which they all descended.
As these cells divide, they specialize. So you begin with a single cell, the egg, the fertilized egg, that has all the information for all the other cells. And as that starting point divides, the 2 daughters that it produces asymmetrically don't have the same capacity. In other words, they've already, at that first cell division, down-regulated some of their genes respectively, so that this daughter is going to give rise to a whole bunch of different cells that'll do particular jobs. This daughter cell will give rise, for instance, to the germline, ultimately the cells that will become the eggs and sperm for the next generation. So you see right from the start, specialization beginning, and then, of course, these 2 daughters divide and give rise to 4. The same thing happens. Those 4 cells now are expressing only part of their genes with respect to the other 3. So you see a process of continuing differentiation where each time the cell divides, it's changing what genes it's going to express, so that ultimately, as you work your way down through the whole lineage to the tips, this cell here, let's say, at cell division 8 in that particular lineage, will only be doing a digestive task, whereas one of its cousins, maybe over here on this lineage, will be a nerve cell. Now, those cells will still have all the DNA that they had at the beginning, but most of their DNA will be shut down and not expressed. They'll only be expressing the genes and proteins that they need for their particular job, which to me is absolutely remarkable. You see a decision tree, sort of like a logical switching pattern, where you begin with your starting point, you've got all the information there, and then that information is subdivided in various ways, parceled out along different lineages, where this group, let's say, is going to be muscles. That's the role that those terminal cells will be playing. This group over here will be only the intestine. This group on this side will be the germ cells, the eggs and sperm and so forth. And there must be some governing logic, some control system that tells those lineages what they're going to do as they are specializing. And I think from the perspective of an undirected process like natural selection or evolution generally, it's very hard to see how you could build that without knowing where you were going. So right from the start, there's a decision being made. This daughter cell is going to give rise to a completely different set of cells than this one. So you're making a decision at that initial point with respect not to these daughters really, but with respect to what they're going to give rise to way down in the lineage. That's a logic control problem. That's an engineering problem. That's where you say, we're going somewhere. This isn't just cancer where you have willy-nilly cells reproducing themselves and, out of control multiplication. No, you have a controlled process of increasing specialization where the first decision is being made with respect to its ultimate target. It defies imagination to conceive of any kind of cause that can do that without knowing where the target is. You've got to know where you're going.
The developmental process of C. elegans poses a mystery for modern Darwinian theory and its undirected mechanism of natural selection. The successful creation of a live C. elegans requires many intermediate cell divisions. Yet the temporary cells created by these intermediate cell divisions play no functional role in the adult worm whatsoever. Instead, they merely serve as stepping stones in a long journey that will eventually reach a functional organism at its conclusion. But natural selection can't select a future function. It can only select features that are advantageous already.
If something's going to function in natural selection, it's got to function now, at this particular moment in time. Not 5 minutes from now, half an hour, a week, 1,000 years. So a process that lacks foresight, in principle, cannot build an unfolding trajectory, an unfolding lineage where you need to know the target. That's the fundamental difficulty for any undirected process of evolution.
What natural selection and other undirected natural mechanisms cannot achieve, intelligent agents can. Intelligent agents are able to foresee distant functional goals. Intelligent agents can coordinate and choreograph the assembly of many separately necessary parts to achieve a functional end.
When I look at animal development, I see a trajectory. It's, in a sense, the quintessential end-directed or teleological process in nature. You're pulling back that bowstring, and you've got a target over there 50 yards away, and you want to put that arrow right in the middle of that target. You need to know what you're aiming at and why, and for that you need a mind. The case for design could not have been made more explicit. I mean, of course, there are much more complicated developmental pathways in nature. A human baby, over the 9 months that it takes to build one, that's something that if we ever come to understand it will really take our breath away. But even these little worms, a millimeter long, sort of humble little creatures out there in the compost heap, they carry the signal of design unmistakably.
So one common attempt to explain the origins of new body plans is to appeal to what are known as Hox genes. And these basically code for what are known as transcription factors. And transcription factors are proteins which essentially regulate the expression of other genes. And they basically control the anterior posterior patterning of an organism like C. elegans or Drosophila. And so the combination of Hox genes that are being expressed regulates the body plan of an organism, such as Drosophila in this case. Drosophila being a fruit fly. And it turns out that mutations in these Hox genes, because it's involved in regulating the expression of other genes, can have really striking anatomical effects. So for example, there's a gene which is known as antennae, and if you mutate it, then the fruit fly can sprout legs where its antenna should be from the head, as you see in the photograph up here. Got legs sprouting from the head where the antenna should be normally, and that's due to a mutation in this antennae gene. There are also other examples. There's another gene that if you modify it, then it results in four-winged fruit fly. And so instead of having the halteres or balancers and 2 wings, now the Drosophila has 4 wings, but those not being anchored to the muscles and the brain is not functional. In fact, the Drosophila is a hopeless cripple and it can't survive in the wild outside the laboratory. So these tend to be disadvantageous rather than advantageous to the organism. Nonetheless, it's caused a lot of excitement in the evolutionary biology community. So, for example, Jeffrey Schwartz of the University of Pittsburgh, he has written that, so we're still in the dark about the origins of most major groups of organisms. They appear in the fossil record as Athena did from the head of Zeus, full-blown and raring to go, in contradiction to Darwin's depiction of evolution as resulting from the gradual accumulation of countless infinitesimally minute variations. However, his proposed resolution to this is to say a mutation affecting the activity of a homeobox or Hox gene can have a profound effect, such as turning larval tunicates into the first chordates. Clearly, the potential homeobox genes have for enacting what we call evolutionary change would seem to be almost unfathomable. Now, I'm personally not convinced by this line of argument, and I'll give a few reasons why. Precisely because Hox genes coordinate the expression of so many other different genes, experimentally generated mutations in Hox genes have proven to be harmful. And furthermore, Hox genes in all animal forms are expressed well after the body plan is established. And Hox genes only provide information for building proteins that function as switches that turn other genes on and off. They don't contain information for building those structural parts. So, in terms of, in the fruit fly, by the time that the Hox genes are actually 6,000 cells have already formed. And the basic geometry of the fly, so its interior, posterior, and dorsal and ventral axes are already well established. And also mutations in Hox genes that do not have all the genetic information necessary to generate new tissues, organs, and body plans. So mutating them cannot possibly generate new forms of animal life. There have also been a number of studies done on different organisms, including Drosophila and zebrafish, which reveal that modification of the early developmental processes actually has catastrophic downstream effects because all subsequent development flows from those early processes. And so this results in another problem of what Dr. Stephen Meyer has called the problem of being major but not viable, and viable but not, or viable but not major. So the proposal that early actin developmental mutations can cause stable, hereditable, large-scale changes in animal body plans contradicts the results of 100 years of mutagenesis experiments in organisms such as fruit flies and nematodes, zebrafish, and mice. And I'm happy to send anyone the literature that deals with these experiments if anyone's interested. So early acting body plan mutations invariably generate embryonic lethals, organisms that don't survive, that is, dead animals incapable of further evolution. So in order to generate a genuinely new body plan, early embryonic changes must take place. Yet any mutation acting early enough to cause a substantial change in body plan always results in a dead embryo. In their Nobel Prize-winning work, Nusslein, Volhard, and Weishaupt observed that early developmental mutants never hatched as larvae. So while these mutants exhibited dramatic changes to their body plans, both in number of segments and their polarity, they were evolutionary dead ends. And of course, dead embryos can't reproduce. So the problem, the dilemma that evolution is confronted with is that early acting body plan mutations, because remember that the body plan is laid down very early in development, right at the start of development, actually before even the Hox genes are expressed. So early acting body plan mutations, the only type of mutations that would be relevant to changing an organism's body plan, are eliminated immediately by natural selection because of their invariably destructive consequences. But later acting mutations that generate viable changes on the feature of animals, these changes don't affect body plan or global animal architectures. So the dilemma then is that major change is not viable and viable change is not major. So it's a catch-22 problem for the evolutionary narrative. Finally, I'll mention the fact that there is information that's relevant to determining the morphogenesis of organismal form that goes beyond the purview of the DNA molecule. And the relevance of this is that if it is the case that much of the information necessary to drive the formation of a body plan does not reside in DNA, then you can mutate DNA indefinitely without respect to time or number of trials, etc. And you'll never be able to generate the type of form and structure you need to build a new organism. It's simply the wrong tool for the job because much of that information actually exists beyond the purview of the DNA molecule. So one source of epigenetic information that affects embryological development is found in the arrangement of sugar molecules on the exterior surface of the cell's plasma membrane. And I'm happy to send anyone the literature dealing with this if anyone's interested. Sugars can attach to the membranes of lipid molecules to form glycolipids, and the cell surface patterns that emerge as a result can be extremely complex and hence have a high level of information storage capacity. The ability of these structures to influence the arrangement of cell types during embryogenesis has led to it being referred to as the sugar code. Another example is electromagnetic fields. So this is another contributor to morphogenesis, and it relates to the distribution and spatial arrangement of ion channels in the cell membrane. Now, although these ion channels are themselves encoded by DNA, their spatial distribution is not. So these ion channels can establish an electromagnetic field which can influence the form of a developing organism. So much of the information necessary for specifying body plan development actually exists beyond the purview of the DNA molecule. So then, finally then, in conclusion, the history of life does not provide adequate resources to resolve the waiting time problem. Cost-cutting reductive evolution is far more prevalent than mutations that build new functions. And the ability of these structures to influence the arrangement of cell types during embryogenesis has led to being referred to the sugar code of the information that exists beyond the purview of the DNA molecule. And that's a problem because mutating DNA is simply the wrong tool for the job for generating new forms of animal or new animal body plans. So I'll close with that. Thank you for your attention.
All right, so let's do like we did last time. We'll open up with a few general questions, and then those that are wanting to stay for a little more technical scientific discussion, we can have more questions there. Feel free to type them in the chat box, or you can open up your mic. I'm going to start with reading one of the questions that is a little more general. There's some more detailed scientific questions that I want to get to, but one of the more general questions is: do we have any evidence from observed microevolution that new molecular or protein structures have been constructed?
So can you, sorry, say that again?
Yeah, it's do we have any evidence from observed microevolution that new molecular or protein structures have been constructed?
Not new fundamental structures, no, we don't.
Okay. And then next question is, how does this account, this is referring to the first part of your talk, for artificial selection happening on very fast timescales, cows, maize, dogs, etc.?
Yeah, so artificial selection is simply breeding organisms for particular phenotypic characteristics that are preferred by the breeder. and generally these involve loss of genetic information rather than gain of genetic information. So, I mean, no one's doubting that natural selection is a real process. So that's not in contention. The question is whether natural selection coupled with chance variation is sufficient to explain genetic novelty. And that is, I think, demonstrably, I think it's demonstrably inadequate to account for genetic novelty.
So you're saying there that these, again, the person that's selecting for, say, a very small dog, Chihuahua or something like that, that seemingly those dogs look quite different from the ancestor that they came from. But you're saying that that's not a good example of illustrating what you're talking about, because there's not novel functionality being created. It's more selecting within existing genomic variants that were already present in the ancestor.
Exactly. Yeah.
Right.
No,
Okay.
basically the question is, it's a response to the two mutation problem. So you have things like maize and et cetera, that you have like dramatic changes over time. There are big changes between these things that are not only one genetic variant, but they're multiple genetic variants. And you see this also in human evolution. You have different people looking quite different over time, and these traits being multiple variants over time. So it doesn't seem that recombination is such an inhibiting factor as you were proposing previously. So you can have these multiple mutations that are being able to be together at once. So it's not too difficult, I would say.
Do you have a specific example in mind where there are demonstrably multiple coordinated mutations that are required to produce a phenotypic effect, and that this has indeed been accomplished?
I think you're just like looking around at the people in this chat. Like we have people with very different things that are different genetic variants that are combined in different features together, that are obviously like large numbers of genetic variants that are together seems to be a answer already.
But do you have examples where you require multiple coordinate mutations, none of which singularly would confer an advantage, but combined they do? Do you have any examples of that?
I mean, I was saying like we have lots of these differences between the people on this chat that have not particularly, some of them are beneficial, some of them are not beneficial, but they're like, if you just look at the different pictures of people around here, we have different ethnic groups varied over a short time period. And those are coordinated mutations of different genetic mutations.
No, I would disagree. I mean, that's just genetic variation. So I don't understand your argument here. That's just genetic variation.
So like let's say, Nathan, let's go deeper on this one. So like, for example, if we use skin color, that's the obvious one. So obviously, if someone is more fair in their complexion, they're going to have more mutations that have accumulated in the melanin production system. But those are generally, I think everybody would acknowledge, those are loss of function, not multiple coordinate mutations that we can observe, at least in the example that you're giving.
Loss versus gain is kind of like based on the environment. So like, it's like, I understand the distinction you're making, but it's not necessarily like an obvious distinction, because there are like, it's just relative to the environment. So like, it's kind of like a difference in function.
Right. So you can make more vitamin D, for example, if you were living in an environment that had, for example, less sun. Yeah, but from a biological perspective, what's that?
Or eye color, et cetera, like lots of these things.
Yeah. But from a biological perspective, if we look at, again, let's use the example of skin color here. The person who's very fair compared to the person who's dark, they just have a bunch of mutations that have knocked out their melanin production system. It doesn't seem like a particularly compelling example of what he was talking about, where you have some whole new system that's come into play, where you have...
Well, you have darker skin color relative to chimps and other things like that. Like you have, chimps have very like light skin color. And
so I think you're confusing simply variation with gain of genetic novelty. Or even, I mean, even something being advantageous doesn't necessarily mean that it's genetically constructive as well. So for example, antibiotic resistance, the most common way of achieving antibiotic resistance is through modification of a target site for the antibiotic, because if the target site is modified, then the antibiotic's no longer able to bind to it. That is advantageous, but it's still breaking stuff. And so it's important not to not to confuse evolutionary advantage with the innovation of novelty.
Right. I'm just saying that innovation of novelty takes much longer, but your fundamental argument is that this is having two mutations that are able to be coordinated is does not necessarily need to be that difficult of a problem if you're able to find similar things in way shorter time frames. These are like 20,000, 50,000 years compared to a million years.
Well, I'm happy to look at examples. If you have a specific example in mind, I'm happy to look at it and evaluate it.
So I actually I responded in the chat, and so I have a specific example if we want to.
Yeah, go for it. Go for it. Go ahead and ask that.
Yeah, so this isn't actually two coordinated mutations, but this is actually a brand new protein.
Mm-hmm. Which paper is this?
So this is a paper. So there's a couple papers. I just put one of them, and so we discovered a bacteria that can process nylon.
Oh yeah, I've written on that. Mm-hmm.
Okay, yeah, and so I'm curious what because obviously nylon is a novel, chemical.
Yeah,
and so this is able to process nylon.
Mm-hmm. Yes. So nylon digesting bacteria. These tend not to, that particular one is a frameshift mutation, which doesn't appear to be a difficult mutation. I've written on that, and as have other people, Evolution News and Views in quite a lot.
Right, but it seems like so when you have a frameshift mutation, you have a totally random new sequence, right? So it seems even more improbable that you would develop a completely novel protein with a totally random sequence that would then be able to process the nylon. And so this actually kind of gets to what I think is was maybe my main thought on this is how do we think about protein domains and structures?
And actually, just for clarification for those who don't know this paper, so what was the original protein doing, and then you?
So I think I don't know what the original protein was doing. I think it's maybe like a transcription factor or something like that. But the frameshift mutation changes basically every residue in the protein.
Right, sure. So it went from a transcription factor, a wild type transcription factor,
right? Duplicate. So it's a gene duplication, and then
And then somebody introduces some insertion of one or two bases.
Right.
And then without doing anything else, this protein can now degrade nylon?
Yeah. Or perform one of the chemical steps necessary to degrade nylon. Yeah. A novel chemical step.
Okay.
And so
do you want to respond to that, Jonathan, before we
Sure.
go to the second question that Nicholas had?
Yeah, I mean, there's quite a lot of posts on Evolution News dealing with this. Basically, so the paper says we propose that amino acid replacements in the catalytic cleft of a pre-existing esterase with the beta-lactamase fold resulted in the evolution of the nylon oligomer hydrolase or nyelinase. So the nyelinase did not have a frame-shifted new protein fold. It was a pre-existing fold with activity that was characteristic of its fold type. So no novel protein fold had emerged in this case, and there was no frameshift mutation that was required to produce nyelinase. I'm happy to send.
Send that to me. Yeah, I'd be curious to dig more into that.
Yeah, I'm happy to send. There's quite a lot I could send you on this paper, so I'm happy to send this.
But I guess that kind of leads me to my second question, thought is, I guess I'll make a comment and see what your kind of reply to it is.
Sure.
And so I'm curious about the protein space. So we talk about the space that the sequence space proteins have to search, or like the DNA has to search. And but I feel like the sequence space is actually not as large. So we have to think about the fact that many different protein sequences can accomplish the same function, right? And we're also moving from one protein to another. And so, there's a lot already in place that, is allowing us to make small jumps with few mutations. So when we do these calculations looking through sequence space, there's a lot of sequence space we can, I think, disregard.
So on the issue of protein folds and sequence space, I'm going to deal with this in a lot more detail, including address that next week. So maybe hold on to that thought, and I'll come back to that one next week.
Okay.
Let me make one announcement, and then we'll transition here. So for those who, we'll kind of wrap up our formal time here, but I sent out an email. If anybody wants to join us, a completely separate group relating to overcoming addictions with a special view on pornography, please send me an email. We've had some preliminary meetings there. It's gone really well, and it's something that I think those of you who have struggled with this or know people who would be interested in, just have either you send me an email or have them send me an email, and we'll add you to the list. We're doing our formal kickoff next Saturday. It's the hour right before society. So feel free to email me there. So with that, anybody who doesn't want to stick around for more technical questions, feel free to jump on. But there's a bunch more questions here that I think would be great to talk about. So another one is, the models also seem wrong because they don't account for models being what you talked about there, Jonathan, for selection happening continually on each generation at differential levels for each increasing change, similar to the artificial selection above. Say there is selection for a larger arm, then each generation there will be selection on the small population size of the larger arm.
Just looking for that question.
The third one, I'm not going in order here, but I was going to do the third one and then the second one. But it's kind of,
you can read the Michael Lynch paper. There's, I think I wrote that later afterwards.
Yeah, there's a link there.
Yeah, on the Michael Lynch's stuff, if that's is that this population genetics model or a different paper?
Yeah,
it is. There's the paper I cited earlier, the Doug Axe paper, on the population genetics model of bacterial evolution is actually a response to Michael Lynch on population genetics. Okay, this isn't the one I was thinking of. I actually haven't read this Lynch paper, so I need to. I can read this and I can give you my thoughts on it if you want at some point.
Yeah, it looks like a fairly technical paper, so I don't know if we can respond in real time here, but yeah, it'd be great to do that. Did you want to say something about this notion of, like, say, a larger arm being selected, that it grows by very small increments, generation over generation, and as opposed to something more stepwise and dramatic?
Yeah, that didn't seem to be a very compelling argument.
That wasn't it was basically that the model that they proposed, they had independent, they're lacking correlative ancestry. There's a bunch of problems with, I think, with assumptions of the model that the other paper that you cited was. I can't remember which paper it was, but like, lack of correlated ancestry between these different groups. There was either they didn't have selection coefficients, or they didn't have like changes in population size that could happen over time with.
Which paper are you talking about? Are you talking about the Dirt Schmidt paper, or?
I think that was that one, but I can't remember.
Yeah. I mean, they're actually criticizing Behe.
No, not that one. The one that they're criticizing, actually. But yeah.
The one that they're criticizing. Are you talking about Behe's book where he makes the argument for malaria? Or are you talking about the Behe and Snoke paper?
I can't remember exactly which one it is.
It's difficult to comment if I'm not sure which paper you're talking about.
Okay. Yeah, never mind. It's just too technical.
Okay. Well, let's go to this question.
But yeah.
Yeah, let's go to the question there from Daniel, which says, can you comment on the role of epigenetic changes, which can also transmit a large amount of information in applying this argument? What are the associated timescales? How valid is the assumption of constant mutation rate? What about second order effects where the mutation rate couples to features of the environment?
So epigenetics basically refers to information at higher level than DNA. Is he referring to Evo Devo or evolution developmental biology? Because that's often
yeah, I'm not sure. I don't know if Daniel, you can comment on that.
Yeah, I'm just asking about the associated timescales for epigenetic changes, epigenetic mutations, because now you have multiple pathways for possible change. How does this play into the model that you're proposing?
So if you're thinking about Evo Devo, I think that it fails for the reasons I outlined in my talk. That the it's generally, I can't think of any developmental change affecting the cis-regulatory regions.
Well,
of
but
oxygen.
I think what he's asking is like, you have now another lever to manipulate, right? So if we think about DNA,
Yeah, a lot more levels actually, right? So how
Right.
does this increased information flow affect the argument?
You mean the fact that we have information beyond DNA, and could that change too? Is that where you're going?
Yeah, they have a much larger parameter space than expected.
Sure, but it's also much larger combinatorial space, which means that you have which means that the probabilistic challenge is even more so a problem than it would be if it was just DNA.
Yeah, in other words, you're saying there, Jonathan, that if the essential argument here is that life exists on a very narrow band of viable DNA space, if we add more dimensions to DNA space, then the viable range becomes even a smaller percentage than what it was already. And so introducing
Right.
new levers or instruments isn't necessarily
Helpful. Correct.
isn't necessarily going to help you. It just makes it, if anything, more complicated
Yeah.
from a timescale perspective to achieve where we're at.
Right. Exactly.
I don't know, Daniel, do you have a comment on that, or is that?
Yeah, just wondering if you could also address the issue of mutation rate, because it's something that I want to understand a bit more. In your models, the things that you've addressed so far seems to suggest that there is a certain unknown number that you try to tease out. I'm wondering how valid the assumption is and what are possible second order effects which might affect it.
I think it becomes even more improbable if once we introduce information beyond the DNA, such as sugar code and the spatial distribution of ion channels.
He's saying mutation rate. He's asking about the mutation rates. This is a different question.
Okay. So say the question again.
So you have your parametrized mutation rate with a single constant, right? How valid is this assumption? What are the
Yeah, I mean, yeah, sure. I mean, all models are simplified, right? So there's no way of getting around a simplification in the model. So you're having to assume an average. But that's the case with all population genetic modeling. You have to make certain assumptions, and your model is going to be more simplified than reality. That's probably why. So Behe actually wrote a response to Jordan Schmidt's paper in 2008, criticizing his work, and he points out that population genetics models, mathematical models, are oversimplified, and whereas his was based on empirical data, which is more likely to be reflective of real life situation. So yeah, I think once you start to think about more real life situations, I think that it's going to be even more challenging than if it's a simplified theoretical model.
Another variant on this question is that if you have a much higher mutation rate, and maybe we don't have all of the same corrective mechanisms for DNA repair as we currently do, and so you're able to explore DNA space more efficiently, more rapidly than today. You know, that people when they're modeling mutation rates in the past are using present mutation rates or observable mutation rates often, not always. And is there room, or how does it affect situations if you have an order of magnitude or even higher mutations entering into the genome, say before we have the ability to correct DNA dimers and a lot of the other things that we now need badly?
Yeah. So the context that I'm thinking of this, so I'm not biologically trained, but I'm thinking of this in terms of, let's say, a machine learning model exploring a very large parameter space. So if you have just a constant jump, a constant learning rate, the behavior is not as optimal as you have a learning rate or a mutation rate in this case, which depends on the environment, where you can get really significant improvements in optimization. So I'm just wondering whether this assumption of constant mutation rate actually inhibits the range of behaviors and the range of optimality that you can see.
You're asking whether constant mutation rates limits the...
Yeah, because if you have a mutation rate which couples the information from the environment, you actually can converge on an optimum much more rapidly.
Right.
But I guess there's more like intuition developed from a large in terms of just classical optimization. For example, I mean, okay, I probably need, this is for a bit more visual, but imagine you're trying to converge to a minimum point in a very large parameter space. You don't want to keep hopping over the minimum point with a constant jump rate. But you instead want to use the information from the environment to inform how you change the system on the next time step. So I'm just wondering if, yeah, like what are the implications of this constant mutation? What happens if you relax this assumption?
I'm not quite sure why you think. Sorry, I'm just not quite sure what the problem is you're trying to highlight.
Okay, sorry. Okay, we can take this offline. If somebody
Okay.
else wants to ask the question. Yeah.
Yeah, feel free to email me, and I can correspond with you.
Yeah, your email is what is your email? Can you give that?
My email is jonathan@jonathanmclatchie.com. You can also contact me via my website at jonathanmclatchie.com.
Okay, so there's a question that you said last time you were going to talk about the fossil record and the flood, and you didn't do that here.
I said last time I was going to talk about the fossil record, which I did in terms of the waiting time problem. In terms of the flood, so I believe that the flood is a regional event or a local event. I don't believe it was a global phenomenon. In terms of biblical support for the plausibility of that scenario, there's numerous examples throughout the Bible where global or regional events are spoken of using this hyperbolic or global universal language. So, for example, in the book of Zephaniah 1, this is God describing his judgment on Judah. He says from verse 2, I will utterly sweep away everything from the face of the earth, declares the Lord. I will sweep away man and beast. I will sweep away the birds of the heavens and the fish of the sea and the rebel with the wicked. I will cut off mankind from the face of the earth, declares the Lord. I will stretch out my hand against Judah and against all the inhabitants of Jerusalem. I will cut off from the place the remnant of Baal and the name of the idolatrous priests, along with the priests, those who bow down on the roofs to the host of the heavens, those who bow down and swear to the Lord and yet swear by Milcom, those who have turned away from following the Lord, who do not seek the Lord or inquire of him. So there it speaks about a local, original judgment on Judah, which is spoken of using this hyperbolic or global language. Also, Joseph's famine is said to have been severe of the whole earth. The whole world is said to have sought audience with Solomon, and so forth. In the New Testament, it says in Acts chapter 2 that there were God-fearing Jews gathered in Jerusalem from every nation under heaven. In Luke 2, the census was taken so that the whole world might be taxed. So we have lots of precedent for that sort of hyperbolic or exaggerative language in the ancient Near East. So I think something similar is going on with respect to the flood narrative in Genesis as well.
Great. Let's take this question here, which is a little bit longer, but it says, I have a more big picture question about a presumption I've heard in these talks and others about the nature of evolution versus intelligent design, which seems to be at least part of the motivation for arguing in favor of intelligent design. And that's the idea that evolution is this unguided or blind process. How can you reconcile the biblical narrative of God being active in creation versus the passive nature of evolution? It doesn't make sense to me that evolution has to be unguided, because if God created evolution and he's all-knowing, then he knew exactly how it could lead to the complexity of life, and that it's not a blind process. It may seem blind or unguided to us because we haven't figured out all the mechanisms, but evolution could very well be God's intelligent design for creating the complexity of life. It seems like we're anthropomorphizing God and assuming that evolution is blind.
Yeah, a few things there. First of all, it is not really my contention per se that God couldn't have used evolution. It's whether he actually did or not. So we have to look at the scientific evidence to determine whether he actually did use evolution. And I think the evidence points against the sufficiency of unguided processes of evolution. Now, in terms of whether, so the question is that it seems like we're anthropomorphizing God and assuming that evolution is blind. Well, typically, theistic evolutionists, or they prefer to be called these days evolutionary creationists, would argue that even though that design is not detectable in evolution, so it's essentially has fideistic tendencies that, and Scripture I think teaches in multiple places that God is revealing his handiwork in nature. Romans 1:20, for example, says that God's invisible qualities, eternal power and divine nature are being clearly seen, being understood to the things that are made, so that men are anapologetos, without an apologetic, without an excuse. So I think Scripture teaches that God has not taken pains to cover up his tracks, as would be required in an evolutionary scenario. If we do propose that evolution is unguided or mindless, which is the standard textbook account of evolution, then I think that it's very difficult to square that with traditional or classical theism, because not even God can direct a process that by its very nature is undirected. And so it's not simply a theological problem, it's a straightforward logical one.
Let's ask a couple more questions and then wrap up here. So there's a question here, very general question I often get from both technical and technical audiences. The knee-jerk response is to say, yeah, it seems improbable, but given enough time, anything can happen. When I start walking through the details, eyes can glaze over. Have you found a particularly compelling and succinct response to address the time solves everything objection?
Yeah, so I call this the Dumb and Dumber objection, after American movie starring Jim Carrey, where he's talking to a beautiful young lady, and he says, what are my chances? And she says, not good. And he says, not good as in like 1 out of 100. And she's like, more like 1 out of 1,000,000. And then he gets really excited and says, so you're telling me there's a chance, right? So we want to, when we're evaluating these sorts of questions, we want to determine not what's possible, but what's plausible. And that's what we do in our everyday life. I mean, if you're trying to solve a murder or other crime in a court of law, well, we're not looking for possible explanations, we're looking for plausible explanations. I mean, anything's possible. It's possible that the accused fingerprints just materialized on the murder weapon. I mean, it's possible. It doesn't violate laws of logic, but is it a plausible explanation? And of course, it's not. So I think that we look for plausibility rather than possibility.
There's a related question here, which is another common question, which is this question about monkeys randomly typing on a keyboard. And I'll actually give a longer version of the question, but the question as it reads is, if a monkey typing randomly will never type up Shakespeare until the end of the universe, what about 100 million monkeys typing randomly on a keyboard? And in fact, the calculations are very easy to do here. So if you just reduce it down to forget about Shakespeare and just pick one line of Shakespeare, take one line of Hamlet, to be or not to be, that is the question. It happens that's 41 keystrokes. You count all the letters there. And if you assume a small typewriter of 32 keys, not as big as most, the probability then becomes 1 in 32 to the power 41, right? 32 keys to the 41 keystrokes power. And if you assume that you have, this is kind of a fun fact, 17 billion galaxies, each galaxy with 17 billion planets, and every planet with 17 billion monkeys on it, and every monkey is typing for 17 billion years at 1 keystroke per second, the probability that any one monkey in all these galaxies and all these planets is less than 1 in 19 trillion. So even to just get one line of Hamlet there, this pre-specified line, it's very difficult to do that. And so the way that a lot of people frame it doesn't even capture the complexity there. It seems like a lot of, at root, what's at the bottom of a lot of this discussion here is exactly what you said about, you said plausible, but you could also say probable from a mathematical or statistical perspective. How do you convert this monkey discussion, or the time kind of heals all wounds, or the time can do anything notion, into something that's probabilistic and in ways that people get it? Is there a way to capture the uniqueness of protein folding space, or the uniqueness of how special it is to find a functional protein amidst the n-dimensional space of amino acids and nucleotides that produce them?
Yeah, I'm going to talk about this in more detail next week, but just briefly, Doug Axe wrote a book called Undeniable, and he has a really great metaphor or analogy in that book where he talks about a noise-seeking robot that's and you're trying that always moves towards the loudest noise in its vicinity, and you're trying to get it to navigate its way through a football stadium, and how there's far fewer there's a relative rarity of football stadiums relative to the space on Earth, and so how probable is it that by chance the robot is going to head towards a football stadium and arrive successfully at a football stadium rather than just get lost because of the distracting noises elsewhere. So when it comes to the monkeys typing on a typewriter analogy, we have to be careful not to mischaracterize evolution because it's not an entirely random process. There is a non-random component, namely natural selection, sexual selection, but there is but to get to those fitness advantages requires a random process. Natural selection only ensures the survival of the fittest, not the arrival of the fittest, and so it only becomes relevant once you've already found those fitness advantages.
Well, Nate has raised his hand here. Nate, please ask your question.
Thanks again for your talk, Jonathan.
Thank you.
So what I'm about to ask kind of came up in the chat, I think. I'm going to turn it into a question.
Sure.
You often hear an objection to ID that goes something like this. ID doesn't make any novel predictions. And because it doesn't make any predictions, we shouldn't take it seriously as a hypothesis.
Right.
What do you say to this form of objection?
Sure. I touched on this briefly last week, but so intelligent design doesn't make any scientific predictions, I think is false, because intelligent design predicts that when we look at, so let's say we hypothesize that something is designed. Okay, what predictions do we make from that hypothesis?
Oh, in that question was also beyond like the Mount Rushmore example. We're talking about like new, like say about bacteria or viruses, or et cetera. Like we're trying to model their new behavior in the environment and et cetera.
Okay.
What would we say?
Sure. So let me finish. So, if we're hypothesizing that something is designed, then we would predict that we would find high levels of specified complexity. And so when we find high levels of specified complexity, that confirms a design inference. So that is a prediction that comes from intelligent design. That specified complexity might take different forms. So when it comes to the protein structures, for example, we might predict that that the ratio of stable and functional protein structures within the vast array of combinatorial possibilities is astronomically small. That would be a prediction that would flow from ID on the hypothesis that protein structures exhibit design. So I think that intelligent design does make predictions in that regard. It also predicts that when we find, if we look at a particular system in the cell, and we're hypothesizing its design, that irreducible complexity might be a prediction of design or something like that. So does that answer your question?
Well, it seems like too, one of the other predictions that people don't give a lot of credit to ID for this, but I actually think it's one that Stephen Jay Gould used to talk about this a lot. So I referenced him in this, but it's this notion that in the ID framework, you would think that biological systems are unstable, degrading, and kind of heading down the path of speciation and sort of the world not doing particularly well. Whereas in evolution, if evolution was what generated all of the diversity that we have in life, shouldn't we be further anticipating more diversity and the tree of life continuing to expand? And again, Gould used to speak about this a lot, that the tree of life is being pruned, as he would say, in our world, and seemingly is unstable, and we're moving towards, when you have the Burmese python that goes into Florida, it just destroys lots of species, and the world that we live in is not as robust as we might likely believe. And it just seems like the overall tendency for homogenization as opposed to diversification is a massive prediction of ID that, like I said, even stalwarts like Gould would say, like, we don't exactly know how to understand that problem. I don't know if you have a comment on that.
Yeah, sure. I agree.
Okay. Kojo, you raised your hand. Why don't you ask your question?
Yeah, my question was kind of similar to, I think, Nate's. So it might have gotten answered a little bit before Nate raised his hand, but I was just curious your thoughts, Dr. McLatchie, on like, if it's not like evolution as a biological mechanism that maybe God's using to drive things, like what are your thoughts on what other biological mechanisms that like might be at work to like explain some of the complexity of life? Like, I think even if you have an intelligent design worldview, one question that still remains is like, well, if it's not by evolutionary mechanisms, what biological mechanisms might God have architected to explain how things work?
Yeah, and there's different views within the intelligent design community on this. Some ID proponents, like Michael Denton or Michael Behe, are sympathetic or advocates for common descent, the idea that all living organisms are descended with modification from a common ancestor. Other ID proponents, such as Paul Nelson or Stephen Meyer or Jonathan Wells, are more skeptical of common descent. So it depends on whether you take a view where God specially intervenes supernaturally to bring new forms of life into existence, or whether organisms are connected by descent, and whether there's a mechanistic explanation that might be fine-tuned. I like the idea that there's some sort of fine-tuned, there's some sort of mechanism that exhibits fine-tuning. Zachary Adair, a colleague of mine in Germany, is trying to argue that. I'm not at this point not convinced by the evidence that there is a mechanism that is sufficient. So I'm agnostic at this point on exactly how the designer created it. I think that's a logically downstream question from the question of whether it exhibits design. I think you can infer design, and then it's a further downstream interesting question as to how exactly the designer did it. I personally think that there's some good evidence for modest levels of common ancestry, but the evidence for universal common ancestry is not that convincing to me. In terms of theologically, I think that often people draw this sharp false dichotomy between special creation and common descent, when actually, once you start to move away from the gradualistic scenario, the boundary between special creation and common descent starts to become rather blurred. I mean, there's various scenarios whereby perhaps God infuses new information into the zygote or the egg cell of the parent in order to bring fundamentally new forms of life into existence, or perhaps I think reasons to believe, take a model where he takes the genome of a previous organism and then modifies it to bring into being a new form of organism. And so there are a variety of ways in which intelligent design could work. Does that help?
Yeah, that helps a lot, actually.
Good.
Yeah, why don't we call it a wrap here? There's still some more questions which I think we can get into, but hopefully next week we can tackle some of those. I actually think some of the strongest arguments in favor of common descent would be things like viral genomes that are found across, say, hominids and humans that exhibit a high degree of homology. Things like vitamin C, broken genes, that sort of thing. I'd love to, if we can, talk about that.
Sure.
In next week's session. So, all right. Well, hey, great session here. Thanks everyone for joining us, and we'll see you hopefully next week.
Thanks a lot, Jonathan. Fantastic presentation.
Thanks.
All right, bye-bye.
Bye.
Bye.